Hey everybody, Dr. Dooms here. I want to give a little bit of a demo on how to use SnapGene, some of its features, as well as to explore some of our plasmid features when we're looking at our plasmids. So this is the activity worksheet that we have, and as part of that, we want to use SnapGene to help you explore the features that are in plasmids. So following along on this worksheet, what do we want you to do? Explore and define the features of the two plasmids below. Obtain plasmids in SnapGene, go to File, Import SnapGene Online Sequences, and then type in the name of the plasmid. And then SnapGene, hover over each feature or double click on the feature to see additional information. Seems simple enough. So I'm going to skip over PET41A. And actually do PSEP4, mostly because I'm a little less familiar with PSEP4. I'm pretty familiar with PET41A. So this will be a little more fun for me. So I'm going to go ahead, get SnapGene open. I want to import from SnapGene Online Sequences. I could also come up here to File and then go Import and then go SnapGene Online Sequences. Either or works. Can type in the name PSEP4. Click on that. Let's see here, preserve from yes. And for the time being, I just want to open that sequence. So I'm going to go ahead, I'm going to import that. That should download. So immediately we get a little bit of information on what this is, synthetic DNA. It's meant to be used in mammalian cells. The bacteria transformation system that we might want to store that plasmid in is unspecified, and it gives you a little bit of a description of what it's used for. So episomal mammalian vector with a CMV promoter and a hygromycin resistance marker for high-level expression in primate and canine cells. Created, modified, sequence author, things of that sort. So I'm gonna blow SnapGene up a little bit to make that a little bigger. I'm actually gonna get rid of the description panel so we have a little more room. And one of the first things I like to do with plasmids is kind of get them into a state of being that I like to see them in personally. Not everyone has to do this, but I like to do it. So one of the things is that sometimes you want to increase the size of your words on here. So if you go to View Font Size, and you can select to make the word smaller, bigger. So I'm actually going to pump up to biggest because that's going to help me to display things a little bit. So you might have noticed that when I did this, this little box up here came in, this little yellow box, and it says one feature not displayed. So keep that in mind when you shift things around. Sometimes some of these might disappear, but they don't actually go away. It's really hard to display them. So because I blew everything up a little too much, it doesn't want to show me everything. So one of the things I'm going to also do now that the words are a little bigger, I like to toggle this button. You can see where all of these annotated boxes right now are hovering above the DNA sequence. I like to have them just place them on the plasmid. It kind of simplifies the diagram for me. The nice part about that is oftentimes it also allows some of your features to bloop back into existence. So when I'm looking at this plasmid, I can see a lot of different things are going on here. And I can see some of the high-level descriptions. I can tell that we have quite a few restriction sites on this vector here. That is what all these like ASI-SI or RGA1 or SFAI, BAMH1, SFI, Hindi3, that sort of thing. So this is how restriction enzymes are shown. If I click on one of these, for example, KPN1, I can come down to the bottom here and sequence, and it will take me directly to where that enzyme is located on the sequence. And so that's one of the features that I really like in SnapGene is that if I'm in the map view, Sorry about that. Don't know why that popped up. If I'm in map view, I can use this to target down to the DNA sequence. So if I highlight something else like this HSV-TK poly-A signal, I can then click on sequence and it takes me directly to where that is, which I find super useful. When we're looking at the DNA sequence, you can tell that it is displaying both strands of the DNA sequence. Plasmids are circular double-stranded DNA. So we're getting to see both of those. And in this case, we can see that our annotation is displayed below the sequence that it is for. So if we click on HSV, it highlights what sequence it counts for. Likewise, if we see this HYGR gene, if I click on that, you can see that it is highlighting, but it highlights a lot of stuff. And this is a protein coding gene, and I can tell that because it also has a translation for it here. So we can see the one-letter amino acid code for the translation. And nicely, like any good translation or most proteins, it does start with a ATG as a start codon. So we have our methionine. And then this, we're traveling all the way down. We can see that it ends in this stop codon right here, which is this kind of white asterisk within a red box. And in this case, TGA is our stop codon. In this case, because this annotated feature was told that it's a protein sequence, it went ahead and it translated it for itself. But if we wanted to try to detect potential protein coding sequences, SnapGene has this handy dandy little open reading frame predictor. And so it will try to find sequences that have a start codon and a stop codon that are of a certain length. And I can change that using translation options. Usually what I do is I just flick it on real quick and see what gets produced. So we can see that it is predicting quite a few different translations, but luckily the hygromycin gene seems to have exactly what it needs to be. This annotation here also looks like it is a full protein coding sequences, as well as this here seems to be associated with this open reading frame. But those also take up space and are a little bit annoying, so I'm just going to go ahead close those down so I don't have to look at as much as I'm going on. So if we go back down to the sequence pane, I can sit here and scroll through everything in here. You can kind of see up here in this upper pane that it's scrolling through also as I'm going through. And so the 0 point of the sequence, and keep in mind it's a circle, so there's no actual 0, but we have artificially set 0 as right before this element, this CMV enhancer. And so SnapGene always picks its like 0 base to be right here at this like north facing position. Orfs came back on. So it will always rotate clockwise from 0 all the way down to the end of the sequence, wherever that is. If we want to change where that point is, you can always pick a different spot on the plasmid, and I believe come up to view and click set origin, and it'll ask you, okay, I want to renumber all the bases starting somewhere else. So if I kind of say here, you can see it rotated everything a little bit to fit in a little better with that. So in this case, that's not super important. You may have noticed in the view, we can also flip the sequence. So it will do a full reverse complement. So if I flip this, It literally just like reverses the sequence. And so sometimes this is nice because sometimes when you're looking at a sequence, when we're thinking in English, we read from left to right. And sometimes it's easier for us to also read DNA sequence left to right too. So some people are a little more comfortable reading DNA sequence in a specific direction according to what kind of elements that they're working on. I'm going to go ahead and undo that because I actually prefer looking at things this way right now until I know more of what I'm doing. Um, so for the time being, I'm actually going to close down all of the restriction enzymes. Right now, it looks like it is displaying unique 6+ cutters. So this will be enzymes that have a recognition site that is 6 base pairs or more. So, and they're unique, which means they only cut at one spot in this plasmid, which is really nice if you're looking for a single cutter to know where that is. But for the time being, I actually want to go ahead and shut that off, mostly because they are taking up a lot of space right now. And so I can do that by clicking on this like Sal1 button. So it takes all those away, and now I can see the sequence a lot easier. Um, so when I'm looking at my sequence, the original reason we want to put this is we want to define what these elements are on this plasmid. So we've already imported it in SnapGene. We can hover over features or double click on them. So I'm gonna go ahead and do that. Whoops, wrong screen. So let's start at the top. So we have a CMV enhancer. So if I just hover over it, you can see that a little bit information popped up. It says CMV enhancer 20 or 282.661 equals 380 base pairs. So basically 282 to 661 are the bases on this promoter that this element spans for a total of 380 base pairs. And then it also has written human cytomegalovirus immediate early enhancer. So if we double click on this feature, what it calls up is other options that we can do to adjust this feature if we want to. So we can see it has a feature name. There are different types of things. Things can be named. Some genetic elements are pretty common, and so they have standard names here already. We can tell it what kind of shape we want to look like. Do we want it to have an arrow on one? Do we want to have arrow on the other end, arrow on both ends? In this case, the enhancer doesn't have a specific direction that we're looking at. I can switch the color if I want to, but again, I don't need to do this. One of the things that I really like are these notes panes here. I can do things like switch this to gene or experiment, gene synonym. It doesn't seem like there's a lot of information embedded into this element already. So I'm going to go back to note. So note here, they tell you what this is. So if I don't actually, if you read this and it means nothing to you because you don't know what human cytomegalovirus intermediate early enhancer is, don't feel like you're alone because I don't truly know what that is either. So I'm going to go ahead and copy this and go to the trusty internet and Google that and be like, what the heck is this thing? Yeah, determines efficiency, transcription, viral replication, and permissive cells, low multiplicity of infection. Interesting. So it seems like the human cytomegalovirus is a virus, which is semi-obvious. And immediate early sounds like it helps in the viral life cycle in the immediately in the early part of infection. So this is something that the virus wants transcribed like right away. And then enhancer, enhancer elements we can always look up too. So enhancer in genetics, ooh, that pops up, nice. DNA sequence that increases the level of transcription of a gene that is located nearby on the same chromosome. Awesome. So that seems really useful information to know, especially if I don't know that. So I'm going to go ahead and copy that, and I'm actually going to add this in. So enhancer equals, nope, equals, goodness, equals DNA sequence that increases the level of transcription of a gene that is located nearby on the same chromosome. So with that information in mind, trying to unbold this because it looks a little ridiculous, but hey. Um, so to complete the worksheet, essentially, when I go here, okay, CMV, go back to our worksheet. So what is said CMV enhancer? So the CMV enhancer is a enhancer sequence from the I do love a copy and paste moment because I hate typing, so I'm actually just going to grab all of this text right now. Throw it in from the human side of megalovirus. It's a sequence from the human megalo. It is meant to help increase our transcription of our gene. Probably. I say probably because we haven't looked at the rest of the plasmid. But if I look at a general overview of this plasmid, I can look at this and say, oh, this enhancer is right next to a promoter. I know that promoters are used to promote transcription. And so this enhancer being right next to this promoter means that it is probably upping the transcription of this promoter. And so this promoter, CMV promoter itself, looks like it is again, the CMV immediate early promoter. So it looks like these are probably related to each other and or might come from the exact site on the virus, in the same virus that this little genetic element was taken from. Promoter, let's see here, used for gene transcription. Now, part of me probably wonders why this promoter. So again, can kind of go over to Google and be like human, just put in Y, see what comes up. Let's see here. Maybe I throw in the word cloning. Okay, enhance. Ooh, SnapGene has its own information. Too bad it doesn't tell me exactly what it is. Hmm. All right. Looks like this is a promoter that's used just for stable expression, um, which is sometimes useful information to know about what you're working on, especially if you want to use that to manipulate something. Um, used for constant gene transcription. I'm not going to go through this entire plasmid, but you can kind of see how you can search and find. So OREP, Epstein-Barr virus OREP replication origin. So as you probably know about plasmids, if we are going to maintain them in their organisms, they need an origin of replication. This is true of our chromosomes. This is true of bacterial chromosomes. You gotta start replication somewhere. So this appears to be an origin probably taken from this Epstein-Barr virus. I suspect that it is allowing for replication of this plasmid within mammalian cells itself. Likewise, we can do something like, Epstein-Barr nuclear antigen 1, also known as EBNA1. So this is a protein coding sequence, CDS coding sequence, yes. So if I wanted to know exactly what this protein does, I could, again, go Google it. But in most cases, SnapGene will have a lot of this embedded information in it. AMPR, AMPR, when I hovered over it, looks like its product is a beta-lactamase, confers resistance to ampicillin, carbonicillin, and related antibiotics. So we have a bacterial antibiotic resistance gene. Likewise, we also have hygromycin resistance right here with this hygromycin B phosphotransferase. And then notice that the protein coding sequence we have, but in order to complete the genetic circuit, we also have to have both the promoter and terminator that are required for this to be properly transcribed. So in this case, it looks like we have a HSV-TK promoter right after or right upstream of the hygromycin resistance gene, and then we have this poly-A signal, which is part of terminator sequences. This terminator sequence is from herpes simplex virus, thymidine kinase, polyadenylation signal. Likewise, looks like the promoter is also taken from another virus, herpes simplex thymidine kinase promoter. So this is the promoter and terminator for a gene that's normally found in a virus, but something that works probably pretty good in mammalian and canine systems, as the description said about this. So when you're looking at plasmids, this is one of the things that you want to kind of explore as you're going through. And so you could open this up, add a note maybe of like virus-derived appears to work in multiple species. And in this case, this does not look like it'll work in bacteria because it's very much a mammalian kind of promoter. So this would be probably for mammalian expression. This product here, this hygromycin resistance gene, will only really express in mammalian cells, while we might have this plasmid inside of a bacteria so that it's easy for us to grow up a lot of the plasmid or manipulate the plasmid. This protein itself isn't really going to be expressed because the bacteria won't recognize this promoter. It's going to recognize bacterial promoters. So this ampicillin resistance promoter, although it doesn't say, is a bacterial promoter that will work for the bacteria. Likewise, if we continue to look at this, I can tell that these elements here, this origin, is for high copy replication in bacteria. If I do a little more Googling, I can definitely go ahead and figure that out. This is something that I know as an instructor. So I can tell that at least these elements seem very bacterial, whereas this origin seems like it's meant for the mammalian system. This resistance cassette right here looks very mammalian. Likewise, cytomegalovirus, very much mammalian. SV40, polyadenylation signal. Wonder what SV40 is. Part of my instinct is that this is definitely something mammalian because the promoter is mammalian, but you never know. Let's see here. SV40, simian vacuolating virus, monkeys and humans. So again, probably something that works pretty well for mammals. And then the last part of this plasmid that I want to draw your attention to is this cute little feature here. If I hover over it, it says multiple cloning sites. And so this is abbreviated as MCS. But this is a site where we tend to have a lot of restriction enzymes located for easy cloning, for restriction-based cloning. So if I go ahead and actually open those restriction enzymes back up, we can see that this entire area has a ton of restriction enzyme sites. If we hover over top of them, you can see exactly what the recognition site, so PDI1, has this cleavage site of GCC, GGC, and you can tell it's a palindromic site. And depending on where the arrows are, you can also tell how it's going to cut the DNA sequence because the two arrows on both of the sequences are right across from each other. This is going to be a blunt-ended cutter, and so it's going to cut straight through. Something else like Hindi 3 is not a blunt-ended cutter. It actually leaves these hanging 5' ends, and you can see that it cuts between the A's on the sequence, both on the top strand and on the bottom strand, which leaves these 4 overhanging nucleotides that we would have in the middle. If I want more information about these restriction enzymes, you can always double click on them and it'll call up this little resource that it has to do with a lot of restriction enzymes. It will tell you the type of overhang that it has. It'll tell you exactly where it's cutting and what orientation it is. I can get information about where I can get it from specific companies. New England Biolabs is a very common place to get enzymes. There are 2 different versions of this enzyme, Hindi 3 HF and just regular old Hindi 3. If we kind of circulate through them, you can tell that the buffers that are used to incubate them are different for them. And so we can get some other information. I can also click through others and just take a look at them. If you are in SnapGene and you just want to look for restriction enzymes in general, which is also a part of said worksheet, if we go a little farther down, we also want you to look up enzymes in SnapGene. You can always go to up here in the enzymes panel. If you click on restriction enzymes, it will call up that same table. And if you click over here and start typing in whatever your enzyme is, you can find the name of it. So whatever, Hindi 3. Okay, so I can find, oh, that was Hindi 2. We want Hindi 3. We could do PVU 1, PVU 2, which are different from each other. So that spelling makes a huge difference. Is there a PUV version? Nope. So you can have some that are kind of tricky, like SCA1, SCA1. Looks like this. It's a blunt-ended cutter. But if we do SAC1, just switch those letters around, we have a totally different enzyme and a totally different recognition site. So you always want to be really careful when you're looking for enzymes and looking at how specific they are. But you can use this to find different enzymes, information about it. The enzyme database is also available, but I find it much less easy to use. So I tend to use the other version of it because I like to simplify. Um, So when you're within a full map like this, there are a couple other buttons at the bottom. You can see that we have a breakdown of all the enzymes that are here. I could do something like clicking off a few of these, and what it's doing is it's just going to hide those. You're not deleting the sequence out, it's just not going to display them on the display. I can always resend that. By clicking on this and having it show all enzymes. Another useful button down here is features. So this just gives you a table form of all the features of everything that's annotated on this plasmid. So we can sit here and go through and just kind of read through a lot of what is done here. The things that I edited and added will appear in here as well. And then we haven't started designing primers yet, but there's a tab for primers if this happens to have primers. There's also this useful little history pane here that allows you to see basically everything that you've done to this vector. I haven't done a whole lot. I really just changed the origin. And when it says origin, it means like the zero nucleotide. It doesn't mean the actual origin of replication. It's really just an orientation to things. Let's see here. Any other fun things? There are a lot of fun buttons that you can try to click and unclick. If I want to just take away a lot of the feature labels, I can take those away. I can get rid of the numbering system because sometimes you don't want the numbers popping up all over the place. You can view it in a linear form if you want to, but keep in mind that this is a circular piece of DNA, so it is fakely linear. They're just trying to show it to you in this different orientation. I tend to like to see things in the orientation that they are. I can go ahead and hide all the features if I don't want to look at those. And then the last thing that I think would be helpful for us to look at would be how to annotate something. So creating your own annotations or modifying established ones is really helpful. But it can be very nice to be able to add your own annotations for your own information. So I could make something up, but let's try to do something a little more useful. Yeah, why not? Go ahead, look at the ampicillin resistance gene, and I'll just do something simple. I want to clearly mark that this ATG is the start codon. I could highlight a lot more, but in this case, I'll highlight ATG. If I come up to features, I can add a feature, duplicate, remove, many, many, many things. But I can go here, add a feature, call this start codon. Start codon is part of a coding sequence. So I'm going to categorize it as a coding sequence. And the direction that the arrow is pointing, I want to point in the direction that it would be translated. So that currently is the correct direction. I'm okay with maroon as a color. And I do want this to translate it. So I could tell that to not translate it and it wouldn't show the amino acid code, but I'm going to go ahead and leave that on. And so when I do that, we have another codon that pops up in here. The other thing that you might notice is that we get this little line that pops up that says in frame with start codon. So what SnapGene will do with some protein coding sequences is sometimes it knows that you want to line up protein coding sequences with each other so that they will be fused together when you translate them. And so it tries to help out sometimes, especially when there is no stop codon where it knows to stop the translation. And so this is just going to say, okay, all of this sequence is in frame. This is the correct set of codons to be using. And then when it stops and finds a stop codon, it will immediately bail and say, oh, if this actually starts translating here, this is where it's going to stop. Because I used the correct stop codon, of course it stops right at the end of the gene sequence. But if I had told it to translate something else, for instance, so I could highlight this whole sequence, add a feature. I can call it a protein coding sequence, even though there's nothing biologically that says that should be possible. I can tell it to translate, and it will do that. It might not like it, but it will do that regardless. And so what this has done is it's pointing out that this is a stop codon, and then it will show me these other codons, and then again, it's just gonna say, okay, this is the frame that I am, and I accidentally selected the exact same frame to be in. So again, this will still stop at the same stop codon. However, I just created something that is biologically not possible because there's no actual start codon that is going to get it to start uh, translating here. So SnapGene is very powerful. You can do a lot of things, but you can also trick it. So you want to be careful when you're doing things like this, that you always check to make sure that you are still sticking to biological rules. So this is fake. So I'm going to go ahead and I'm going to, I can right click on it and remove the feature. And like it says, the DNA sequence will not be affected, so it's not going to delete the DNA sequence itself. It's just going to remove that annotation. I can highlight DNA sequence and hit delete, and it will get rid of those base pairs for me if I want to. But keep in mind, whenever we're manipulating DNA files and things of that sort, you have to make sure that what you're doing is something that would happen in the real world, like something that you are actually capable of doing, because otherwise you're just reorganizing letters on a page. But we are doing all of this like planning and looking to do for something that is going to be biologically relevant and biologically possible. Um, all right. So I think that's all I want to say for the time being for kind of using SnapGene, looking at features, doing a little more digging on features, as well as looking at some of our digestion sites that we have on here. And I would like to wish you a good day.